Dr. Asrar B.Malik from University of Illinois at Chicago, and he would talk about signaling pathways regulating endothelial barrier function.
Good, thank you. Yeah, I forgot my credit card, so.
We'll work something out.
Thank you for the, thank you for the invitation. It's a wonderful symposium. I especially like that bit about Galen, and it's so delightful to see Bill Aird so excitable when he talks about Galen. I think he's probably the only one. So, so I'm going to be talking about, the sort of the intimate mechanisms that regulate permeability, and more in terms of what little we know, and I guess some aspects of what we know, and perhaps areas that are getting a bit overworked. So, to begin with, I think it's good to talk about a summary of the field, or get a general sense of the field, as I've tried to deconstruct it here. So, the main aspect of permeability regulation is really at the level of the junctions. By that I mean adherence junctions, and these are well known to be consist of a series of sort of interconnected. Can you hear me? Interconnected proteins. These are weak adherent, which is very specific to endothelial cells. Then another one, a family of catenins, P120 catenin, beta catenin. So it forms this network of these junctional proteins that are then connected to the cytoskeleton. So it's a very important arrangement, and this arrangement here is really central to the dynamic regulation of barrier function. We've shown that, and multiple other groups have shown that. But there's some important aspects of it that I think have been recent in their understanding. This concept of a disassembly of adherence junctions is a fundamental concept because, I like to think of junctions as plastic, in that they can disassemble. I don't mean they rip apart, but I think this is a very ordered a mechanism. It's a very orderly mechanism, and they also have the capability of reannealing or reassembling junctions on the basis of certain signaling cues. And here I've kind of diagrammed what would happen if the barrier function was disassembled. There would be a drop in what we measure. We can measure actually is electrical resistance. So there's a drop in that, and these are pretty canonical changes, and this may vary according to the stimulus that disrupts junctions, and eventually, in this case, it's 2 hours, but it may be 4 hours, it may be 6 hours, and so on. But depending on the initial stimulus, there's this reassembly or this restorative phase, and so the two unknown questions, in my view, which I think should be studied in all microvascular beds, is the nature of the disassembly. One important aspect of disassembly is that weak adherence is actually internalized. It's an active event. It's endocytosed. I know endocytosis is not a verb, but it's literally endocytosed on the basis of some specific signaling pathways we recently described, and then there's a return to the membrane where it reassembles and reanneals the barrier. So this weak adherent internalization event is a discrete event, and I'm going to show you that it's controlled by certain signaling pathways. Then the reannealing itself is again a event on some specific signaling pathways, and there are a couple of them because it's a complex process and it's a physiologically, homeostatically relevant process. So one of them involves RAC mediated trans interaction. Remember, weak adherent binds in a cis manner, but it also binds in a trans manner. So it's RAC, this GTPase RAC mediates this. And this other GTPase, CD42, partitions this function of these Rho GTPases, and its function is to elicit by some active mechanisms formation of lamellipodia, which I'll describe for you. So this is the, what we've shown this is to be essential, this protein kinase C-alpha regulation of V-cadherin, and it functions in a way, I think, in all vascular beds. Basically, you have this domain of P120-catinin, which is a binding protein. It's very perimembranous binding protein, right at the edge of this juxtamolecular domain. PKC phosphorylates it, so there's phosphorylation at this serine domain. PKC then, this phosphorylation dissociates it, and this protein AP2, which is an adapter protein for clathrin, binds there, and then you have this clathrin-mediated internalization of V-cadherin, and hence the disassembly of junctions. So it's a well-regulated, well-coordinated process. Now, what I think that from a perspective of how do we restore integrity, I think that's a very important question for all inflammatory diseases in the heart or the brain or wherever. So what regulates this sort of re-annealing or reassembly process? And again, one can sort of partition various signaling factors. So one of the critical ones is CD42. Now CDC42 functions by inducing polymerization in these newly formed lamellar protrusions, and really allows these cells to meet one another and to contact one another, and you can visualize that, and various studies, some of them shown here, indicate that CDC42 is a requirement, not only a requirement, it's sufficient for the formation of lamellipodia protrusions and for this initial event, or this initial step in the formation of adherence junctions. Now, because of the lack of knowledge, we don't know events 1 to 10. We know events 1, 2, and 4. But so there are probably a series of handoffs from this initial event to the subsequent event that leads to the formation of these adhesive junctions. Now, we've probed somewhat in the role of RAC1, which is the other interesting GTPase in this context. And the approach we've taken is that we can photoactivate RAC locally at the junction using these are collaborations with Klaus Hahn, where RAC can be photoactivated using in live cells using this expression of this construct. And when you apply UV light, this unfolds, and so you have activated RAC at the level of the junctions. So in this case, at least, we use the photoinsensitive probe, which is this one, and then well, I'm sorry, this is the dominant negative one, and these are the two controls, and this is the actual probe we used. So the question we asked was, what is the consequence of localized activation of RAC in promoting reassembly? And this is a complex graph, but I think it makes one or two simple points. And that is, if you activate RAC at the junctions here, this is where the UV light is shown, and you look at mobilization of V-cadherin at different times very rapidly. So within 3 to 4 minutes, you get increased fluorescent intensity of V-cadherin at the junctions. Whereas if you use the inactive probe, photoactivated probe, there's no increase in V-cadherin. So what this means is that RAC functions, and it only is a function in mature junctions, junctions that already have adhesion complexes, that RAC functions in actually making the barrier stronger, because it enables the accumulation of V-cadherin at the junctions. So RAC is, and again, like CD42, which is an early step, RAC1 is a subsequent step that promotes the accumulation of V-cadherin at the junctions. And through many studies, we came across a sort of general model or general concept that RAC stabilized V-cadherin trans-interaction to reduce the V-cadherin dissociation. So the accumulation is the function of the reduction in V-cadherin dissociation. And this is because RAC functions by inhibiting Rho-A. We know this cascade, inhibiting Rho-A, which thereby inhibits myosin light chain kinase phosphorylation and reduction in tension. So the reduced tension occurring at this junctional domain is the primary factor responsible for RAC-induced assembly of the junctions. And this is clearly an important point, we think, that because it's a way of stabilizing a junction, and it's a way various junctional stabilizing mediators such as sphingosine 1-phosphate function. So this has important relevance for trying to understand the biology of junctional re-annealing. So a few words about the other pathway. The pathway I've talked about so far is the paracellular pathway. There are a couple of people in this room, for example, Radustan, who know quite a bit about this other pathway. So this other pathway is important, but a bit of a Cinderella in that much less is known about it. But we do know it's important. We don't know why it's important. We know it's important. And this is the caveolar pathway. If you put albumin in the luminal side, this is small particles of albumin, 1 or 2 nanometers. So albumin particles are engulfed in these caveoli and are transferred from the lumen to the basal side. So this is the basal side here. This is an alive animal, and you can see a caveola depositing these albumin particles. There's one out here. This is only 5 minutes after adding these. So it's a very rapidly activated system, and it's a fundamental system for transport of large molecules across the endothelial barrier. Please, you can see here that if you look at the junctions, there's no albumin nanoparticle. Junctions are simply too small. The diameter radius is too small for the transport of these molecules. So albumin and other macromolecules, at least in these type of endothelia, and clearly in the blood-brain endothelial, can only be transported through a caveola, but that's another question. So this is an important pathway. So if you want to play sort of genetic games with this pathway, for example, if you knock out a protein that is responsible for caveoli formation, caveolin-1, in endothelial cells, you can see interesting compensatory mechanism being activated. And you can see here that in fact there is transport, across the adherens junctional complex here in the absence of caveolin-1. And this shows another very dramatic picture, also in the absence of caveolin-1. You can see albumin being transported by this mechanism. Am I over time yet?
Just to wrap up, you have 1 minute.
Wow. Okay. So there's an interesting compensation occurring. And the compensation is this. This just appeared recently. And that is there's a consilience between the caveolar-mediated pathway and the adherens junction. So when caveolar pathway, there's an involvement between the caveolar pathway, release of eNOS, nitrating this Rho gap, P190 Rho gap. So this is a nitration reaction, which kills the gap activity. So you have Rho activity at the junctions, and then Rho opens up the junction. So there's a very interesting crosstalk between these two pathways. So my final bit, just one or two slides, is that we've recently begun to address the question, what is the mechanism of endothelial cell repair? This has nothing to do with renewing, but it's repair. So we made this, I think, an interesting mouse model where we have tomato red mice, tomato red as a fluorophore labeling non-endothelial cells, and GFP labeling endothelial cells. So we can then test the idea of non-endothelial cells being labeled red and endothelial cells being labeled green. So the question we wanted to ask, if you injure the endothelium precisely, do other cells take over the endothelial barrier or endothelial function? By other cells, I mean red cells, let's say progenitor cells from the red tissue, or do endothelial cells themselves induce the repair process? And the long and the short of it is, and this just shows the model, so you can green label the endothelial cells, everything else is labeled red. And the long and the short of it is, if you induce LPS injury, let's say, of the endothelium, there's a decrease in the amount of GFP at 24 hours, but importantly, recovery, so indicating full recovery of this injury, and we titrated the dose, et cetera, to arrive at that. Now, what this means is that this is due to a decrease in the green cells or the endothelial cells. It's not due to increase in the red cells, because if that had occurred, this wouldn't have recovered to normal. So what this suggests is that they're facultative endothelial cells. That is, endothelial cells that the green cells that are promoting repair.
Move to the last two. Please.
Sure, that are promoting repair are specialized, and they have some specialized functions, and they're facultative. So the unknowns in this area, there are many, and I haven't learned many lessons, such as keeping on time. So one of them is the plasticity of endothelial cells. The other is the notion that you can close adherence junctions. The third is that trafficking of caveoli are important but poorly studied. Endothelial lineage analysis, I think, is a way forward. And gaps, there are a lot of gaps. Details of the signaling pathway, the precise working of the transcytosis pathway, definition of facultative endothelial cells, and so I mean, this is a very interesting idea. How does liquid and solute, how do they transport across, as well as blood cells and cancer cells? So I'm making this kind of heretical argument. Metastasis is really a disease of the endothelial barrier. Thank you.
We're happy to take questions. Just one or two quick questions,
Or
please.
half a question.
And you better be prepared to buy a lot of drinks tonight.
Okay.
Can I start?
Yes.
Yeah. Hi, Marty Kluger from Yale. That was a very nice talk, and I think you're absolutely right that the localization using the photoactivatable RAC1 is a technological improvement over looking at RAC1 in a global way. But you also spoke about maturation of junctions, and I wanted to bring up the point that tight junction molecules are expressed throughout the microvasculature, and more so, say, in capillaries than post-capillary venules. So I think that some of the mechanisms you discussed about reformation and also taking junctions apart should be applied to those molecules and the different caliber beds.
Right. It's a very good point.
Yeah, next question. One more. Okay. Very quick.
Yes, this Irobo Bogu from University of Alabama in Birmingham. I want to first of all find out which cells you are working with, which endothelial cells we're working in particular, because I guess the ability to form adherence junctions and tight junctions will depend on the cell type you're working with.
Well, actually, we use the ones where we can get adherence junctions. So we don't use the cells. Microvessel cells, for example, microvessel endothelial cells, they form very nice adherence junctions.
And did you look at tight junctions at all?
Tight junctions are very in our experience, are not that well expressed in the endothelium, except for the unique brain endothelium.
Okay.
And the main junctional permeability pathway, after much hair pulling and this and that, appears to be the adherence junctions.